It's a pleasure to be here, and it's a pleasure to be here in New Zealand for the first time. I am from Chicago, although not originally, as you may hear from my accent. I'm a practicing clinician, have taken care of transplant patients for the last 20 plus years, and can have a lot of patients, and my understanding is that most of you have gone through transplants or are relatives of patients who have gone through transplants and may have a number of questions. So when preparing this talk, it is easy to think of a number of issues. It's not that easy to address all those in a talk of about an hour, and one has to make a selection. Any questions that may come up in the next hour or after the next hour, feel free to interrupt me, feel free to ask questions. I don't know whether my talks and my slides here will address the questions that you have, but there's plenty of time for exchange. What I thought I'd do is give you a brief overview of where transplant stands, from kind of a general idea what are the indications for allogeneic, for autologous transplant, and then talk a bit about complications and about how we address complications and what are some practical issues affecting that. Without further ado, so obviously two types of transplant. Allotransplant, where a patient undergoes chemotherapy with the idea of eradicating a disease, or in rare conditions such as aplastic anemia, of preparing the patient for then receiving a transplant from a donor. And there's a number of issues about this donor. Who is this donor? The classic donor for an allogeneic transplantation is a sibling, as probably many of you know. But not everybody who has a sibling who has the same bone marrow type. What determines whether a sibling matches is that the sibling has the same HLA type, meaning HLA type is human lymphocyte antigens. So there are a number of genetic characteristics that make us similar and that make us accept a bone marrow from one another. And it's clear, you look at a brother and a sister, they look like one another, and people that are not relatives don't look like one another. But so that there's genetic similarity. There's many more genes shared between siblings than between others. Some are more important than others, and the most important ones by far are these genes called HLA, or the major histocompatibility complex. And we talk about HLA-A, HLA-B, HLA-C, HLA-DR, DP, DQ, a set of 8 different genes, one inherited from each. We have each of them in double, one inherited from father, one inherited from mother. And to be a good donor for one another, one has to share the same HLA-A, the same HLA-B, and so forth. For siblings, it can be reduced to a much more simple problem, in the sense that, and again, I have to talk a little bit about biology here, but I think it is important. We have 46 chromosomes. All these genes are encoded for on chromosome 6. So if you get the same chromosome 6 from mother and from father, then you will have the same HLA type as your sibling. So here I've color-coded it. Mother has a light blue, dark blue, father has a red and a yellow. This child has received a light blue from mother, a red from father. The 5th child here has received exactly the same chromosome from father and mother, and therefore is HLA identical. It's 1 out of 4 that we will get the same type. It's also 1 out of 4 that we will receive completely the same, completely different type. So this sibling has received blue, and red. This sibling has received dark blue and yellow, completely mismatched. It's 1 out of 4. About half of the siblings are going to be half matched. They have received the same from father, but a different one from mother, or the same from mother, a different one from father. This also tells you that parents are hardly ever completely matched with their children. The parents are half matched with their children by definition. It is rare, not completely unheard of, but it's rare for parents to match their children. So this is the big, the big issue when we do an allogeneic transplantation. Does the patient have a donor? Does the patient have a donor in their family? It's 1 out of 4 for each of their siblings. Donating marrow is, or stem cells is these days, I'm not going to talk much about that unless somebody has a question about the donors, but essentially donating marrow or stem cells these days is a safe procedure, something we get asked about all the time. What is this going to do to my brother? What is this going to do to my sister? Donating stem cells. What I tell my patients is it's safer to donate stem cells than to drive through Chicago at night. And it's like, it's a big deal, but it's not such a big deal. Now, where do we use indications? Where do we use allogeneic transplantation? I'm not talking right now about autologous transplant. Some of you may have undergone an autologous transplant, which is using patients' own stem cells. We'll talk about that a little bit later because it's a very different procedure. But allogeneic transplant in the States, 6,000 a year. This is what and what do we do it for? Mostly for acute leukemia. AML, acute lymphocytic leukemia, acute myelogenous leukemia, the green bars, myelodysplastic syndrome are the 3 big indications these days in the United States and probably worldwide. If one would have come 10 years ago, 1996, it would have been much more commonly chronic leukemia, chronic myelogenous leukemia. For chronic myelogenous leukemia, about 8 years ago, a drug was developed called Gleevec or imatinib that has revolutionized the treatment, and it's become rare to do allogeneic transplants for CML because we don't need it very much anymore. Lymphomas occasionally, although they undergo more autologous transplant, and then other diseases such as aplastic anemia. And I honestly don't know how common that is here in New Zealand. I know in Asian countries this is quite common. In Far East Asian countries this is quite common illness. In the States this is unusual disease.